Before we explain the process of protein synthesis, we are going to describe a protein first. So proteins are macromolecules and they're made of amino acids, which are monomers of proteins, and the polymer is a polypeptide chain. So proteins are held together by peptide bonds, and an example of a protein found in vertebrates is hemoglobin, which is an iron-containing oxygen transport protein in red blood cells. So it simply just carries O2 throughout the body.
So there are three main types of RNAs which aid in transcription and translation. The first type is mRNA, or messenger RNA, which results from transcription and is due to the copying of information from one molecule of DNA to the other, which is mRNA. mRNA is the basis for building proteins and the primary structure, and it attaches to the ribosomes in the cytoplasm. The second type of RNA, or ribosomal RNA, is responsible for catalyzing the joining of tRNA and mRNA, which causes the production of the polypeptide chain. rRNA is made up of two subunits, the small subunit and the large subunit. Both of the subunits are made in the cytoplasm by proteins and then exported by the nuclear pores into the cytoplasm. They're only functional when both of the subunits are joined together, and rRNA is the most abundant type of RNA. A significant difference between eukaryotes and prokaryotes is the difference in rRNA. In eukaryotes, the rRNA is slightly larger and has a different molecular composition. Different drugs can have different effects in the prokaryote ribosomal RNA and in eukaryotes ribosomal RNA. For example, the drug tetracycline can inactivate the protein ribosome in prokaryotes because of a different structure, but it does not affect the ribosome in eukaryotes because of a different molecular composition. RNA has a P site, which holds the tRNA carrying the growing polypeptide chain, an A site with an amino acyl tRNA site. rRNA also contains the E site, which is the exit site, and this site releases the tRNA into the cytoplasm. rRNA holds the tRNA and mRNA and catalyzes the formation of the peptide. As the polypeptide elongates inside the rRNA, it approaches the exit tunnel and then is released into the cytoplasm. Research suggests that rRNA and not protein is responsible for the function and structure of a ribosome. Therefore, rRNA is crucial in transcription and translation.
So there are two types of ribosomes, free and bound. Free ribosomes are located in the cytosol, and they're responsible for most of the protein production. And usually the proteins that are produced here are for cytosol function and usually work in the cytosol. And an example of a free ribosome would be enzymes that catalyze the first steps of sugar breakdown. So another type of ribosome are bound ribosomes, and these ribosomes are attached to the outside of the endoplasmic reticulum, or the nuclear envelope. And so these ribosomes produce proteins that are going to be either inserted into membranes, packaged within organelles, or they're going to be exported within the cell, so basically secretion. So an example of this would be cells of the pancreas, because they contain a lot of bound ribosomes, because the pancreas produces digestive enzymes which are usually transported out of the pancreas into the digestive tract.
The third type of RNA is tRNA, and the main function of tRNA is to transfer amino acids from the cytoplasmic pool of amino acids to a ribosome. tRNA is used repeatedly for designated amino acid and carrying it back to the RNA. It consists of the shortest amount of nucleotides from all the RNAs, and it contains hydrogen bonds which allow it to fold upon itself, forming a three-dimensional structure like a cloverleaf. The first end of the tRNA contains the anticodon, which binds to the mRNA codon. The other contains the site in which the amino acid is attached to it. A very crucial enzyme during translation is the amino acyl tRNA, which catalyzes the attachment of a specific amino acid to its designated tRNA. This amino acid tRNA is crucial because without it, we would have random amino acids attaching to different types of tRNA, which would inhibit translation and cause a different polypeptide sequence to form. The amino acid tRNA contains a particular active site for the specific combination of the amino acid and tRNA. The first part of protein synthesis is transcription, which is when the DNA molecule is transcripted to mRNA. This occurs in the nucleus and uses the nucleotides adenine, cytosine, guanine, and uracil instead of thymine, which is used in DNA. Transcription occurs in 3 crucial steps, which is initiation, elongation, and termination. The first part of transcription is initiation, which begins after RNA polymerase binds to the promoter region on the DNA. The promoter region on the DNA consists of the transcription start point, which is a set of nucleotides which initiates transcription and determines which of the 2 strands will be translated. Which of the 2 strands will be translated is determined with the Tata box. The side which has the most nucleotides consisting of the adenine and thymine on the 5' region of the DNA will be transcribed. In eukaryotes, transcription factors are the collection of proteins which mediate the binding of RNA polymerase and the initiation of transcription. The bound transcription factors to the RNA polymerase is known as the transcription initiation complex. Once the initiation stage is over, the elongation stage begins during transcription. In this stage, nucleotides are added to the 3' end of the growing RNA molecule. What initiates this stage are RNA polymerases, and on one single gene, many RNA polymerases may bind and begin to add nucleotides to the 3' end. Another difference between prokaryotes and eukaryotes is the stage of termination. In prokaryotes, the transcribe terminator causes the release of the polymerase from the DNA, resulting in a mRNA readily available to use. Therefore, the mRNA can diffuse to any part of the cell and is already available to use. However, in eukaryotes, the pre-mRNA is separated from the RNA chain, and however, the RNA polymerase II continues to transcribe the cleaved pre-mRNA. When the polymerase reaches the polyadenylation signal AAU-AAA, the pre-mRNA is released from the polymerase, which is then modified.
In this picture, RNA polymerase is adding complementary bases to the strand of DNA to form an mRNA, and this mRNA will eventually travel outside of the nucleus and into the cytoplasm into the next stage of protein synthesis called translation.
A key process in transcription is mRNA processing, which occurs only in eukaryotes. During this process, the primary transcript, which is pre-mRNA, the product of transcription, is spliced and is processed in a separate compartment of the nucleus. The non-coding regions of the primary mRNA, the introns, are removed by RNA splicing. RNA splicing is done by proteins containing special RNA, for example, snRNA, or snRNPs, which contain snRNA and aid in the binding to mRNA. The complex of snRNPs form a spliceosome, which splice the introns out. Guanine nucleotides are known as the G-cap, and the adenine nucleotides are known as the poly-A tail. The G-cap and the poly-A tail aid in the export of the mRNA out of the nucleus and also protects it from degradation during transportation. These regions are not translated into proteins. A key RNA used during transcription is RNA interference, or RNAi. RNAi is a process used to inhibit the expression of certain genes. This process uses synthetic double-stranded RNA molecules which match the sequence of a particular gene, resulting in the degradation of the mRNA. RNA processing is done only in eukaryotes, and the evolutionary significance of that is that it produces variation by joining different sets of exons, and this is seen in the antibody creation by B cells in the immune system. And RNA processing also creates different proteins as the DNA is altered, and the proteins have different structure, therefore resulting in different functions used in various parts of the body.
So after transcription, the mRNA molecule that is produced exits through nuclear pores in the nucleus and then makes its way into the cytoplasm to a ribosome. So to have a brief overview of translation, the point of translation is to create amino acids, and then amino acid chain, and then this process occurs in the cytoplasm at a ribosome, and this process includes tRNA and mRNA, and its crucial steps include initiation, elongation, and termination. So initiation starts off in the small rRNA subunit binds to the mRNA. And after this, an initiator, tRNA, with the anticodon, usually UAC, binds to the codon AUG, which begins transcription. And so a large rRNA subunit binds to the present subunit, and then the binding of the translation components are caused by initiation factors. So these are just group of fat proteins and GTP. So the next step of translation is elongation, and elongation begins with codon recognition, which is when the anticodon of a tRNA binds to codons on the mRNA on the A site. And so the next step is peptide bond formation, and this is when the subunit on rRNA causes the formation of a peptide bond between the amino acid in the A site and the carboxyl end of the growing peptide in the P site. And then after this is translocation, and tRNA is moved from the A site to the P site, and then to the E site from which it is released. So this is a picture of translation, and basically what is happening is the mRNA is in the ribosome, and a tRNA is with its anticodon is bonding with the mRNA, and then the amino acids are on top. Okay, so post-transitional modification occurs after translation, and it's basically a process just to, there are extra steps to fully complete protein synthesis. So these modifications play a role in creating a heterogeneity in proteins and help proteins become the right structure and carry out certain cell functions. And this process occurs in the peptide terminus. So basically there are a few different types of post-transitional modification, and one of these is glycosylation, and this is when a carbohydrate is added to a protein, and this mainly occurs in eukaryotic cells. It occurs when a glycosol group is added to certain amino acids such as asparagine and hydroxylysine. Another type is methylation, and this is when a methyl group is added to either lysine or arginine. Phosphorylation is when a phosphate group is added to either serine, tyrosine, threonine, or histidine. Acetylation is when an acetyl group is added to a protein. And alkylation is when an alkali group is added to a protein such as ethyl. So operons are used only in prokaryotes, and operons act as a gene regulation and are significant in an unstable environment. They also can induce or suppress genes depending on repressors and inducers, which are stimulated by the unstable environment. Operons are a group of bacterial genes and a promoter that help promote the transcription of specific genes. When an operon is transcribed, a strand of mRNA is produced. A promoter is the site where RNA polymerase attaches, and an operator is the region of DNA on the operon that interacts with the regulatory protein, which controls the transcription. There are certain molecules that can either turn on an operon or repress it or prevent transcription from occurring. So inducers are the proteins that turn on or induce the expression of certain genes, and repressors are regulatory proteins that bind to the operator and prevent transcription of structural genes. An example is the TRP operon. When tryptophan is not in the environment, a repressor is made and therefore can't bind to the DNA to block transcription. RNA polymerase then binds and transcription occurs. A second example of an operon is the lac operon, and it is controlled by two regulatory proteins, the catabolite activator protein, which it binds to the activator binding site and facilitates transcription, and the lac repressor protein binds to the operator and blocks transcription. So, and the specific protein that works in the lac operon is allolactose. In prokaryotes and eukaryotes, RNA polymerases are different. So in eukaryotic RNA polymerases are aided by transcription factors. Both have different ways in which transcription is terminated, and both prokaryotes and eukaryotes contain slight variations of ribosomes. Prokaryotes have no compartments in their cell, thus transcription and translation occur concurrently in the same gene. The newly made protein is able to transport efficiently to where it is needed as long as there's no segregation in the prokaryotic cell. However, the eukaryotic cell, the separation of the nucleus from the cytoplasm, allows processing to take place, which does not occur in prokaryotes. And this additional state aids in the regulation and coordination. So, in eukaryotes, eukaryotes also contain a crucial mechanism allowing it to transport proteins from one organelle to another. Prokaryotes also do protein synthesis different from eukaryotes. 1. Prokaryotes do not have a nucleus, so transcription and translation occur in the cytoplasm. As for prokaryotes, they don't do post-transitional modification. So most of our hormones, such as progesterone and insulin, are made of protein, and we need proteins for enzymes, and without enzymes, our bodily functions that normally occur quickly would happen a lot slower. And so protein is an essential molecule in bones, cartilage, and elastin, and proteins are necessary for movement. So more on the evolutionary significance. The genetic code is universal and shared by many organisms. An organism with the same codon will translate the same amino acid, and similar genomes aid in showing common ancestry. The significance of codes is similar to a common language shared by all living organisms and has been present in all common ancestors of modern organisms. It portrays the relationship between all life on Earth.
There are three types of main RNA which are used throughout transcription and translation. The first one is ribosomal RNA, or rRNA, which is only functional when both subunits are joined. It contains a P site, or a peptidyl tRNA site, which holds the tRNA carrying the growing polypeptide chain. It has an A site, or amino acyl tRNA site, which holds the successing tRNA. And it has the E site, or exit site, which releases the tRNA into the cytoplasm. The ribosomal RNA holds the tRNA and mRNA and catalyzes the formation of the polypeptide chain. The second type of RNA is mRNA, or messenger RNA, which is the result of transcription due to the copying of information from one molecule of DNA into another, mRNA, and it is responsible for the primary structure of proteins. The third type of RNA is transfer RNA, or tRNA, and is used repeatedly for designated amino acid and carrying it back to the rRNA. The hydrogen bonds in the tRNA allow it to fold upon itself, forming a three-dimensional cloverleaf structure. One end of the tRNA contains the anticodon, which binds to the mRNA codon, and the other contains a site in which the amino acid is attached to it. Transcription is the first part of protein synthesis and produces the mRNA. It occurs in the nucleus and consists of three crucial steps, which is initiation, elongation, and termination. The synthesis is in the 5' to 3' direction, with nucleotides being added to the 3'. Initiation begins after RNA polymerase binds to the promoter region on the DNA. The promoter region consists of the transcription start point and determines which of the two strands will be translated based off of the Tata box. Elongation is the second stage of transcription. In this stage, it continues to grow and add nucleotides to the 3' end of the growing RNA molecule. This stage is very efficient because many RNA polymerases can transcribe a single gene. Termination differs from eukaryotes and prokaryotes. In eukaryotes, the pre-mRNA is separated from the RNA chain. However, RNA polymerase II continues to transcribe the cleaved pre-mRNA. When the polymerase reaches the polyadenylation signal sequence, the pre-mRNA is released from the polymerase. A crucial step in eukaryotes after transcription is mRNA processing. In this event, the primary mRNA with coding and non-coding regions is processed in a separate compartment of the nucleus. The non-coding regions, or introns, are removed by RNA splicing. RNA splicing is done by the complex of snRNPs, which form a spliceosome. Once the introns are removed, the exons are spliced together. Additionally, enzymes add modified guanine nucleotides to the 5' end and add adenine nucleotides to the 3' end, which is known as the poly A tail. This aids in the export of the mRNA out of the nucleus and protects it from degradation. These regions are not translated into proteins. There are two types of ribosomes used throughout transcription and translation: free ribosomes and bound ribosomes. Free ribosomes are located in the cytosol, and they produce proteins to function in the cytoplasm. Bound ribosomes are attached to the endoplasmic reticulum or to the nuclear envelope. These ribosomes produce proteins that are going to be either inserted into the membranes or exported from the cell by secretion. RNA interference, or RNAi, is a process used to inhibit the expression of certain genes. This process uses synthetic double-stranded RNA molecules which match the sequence of a particular gene, resulting in the degradation of the mRNA.
Operons are a group of bacterial genes and a promoter that help promote the transcription of specific genes. When the operon is transcribed, a strand of mRNA is produced. The promoter is the site where the RNA polymerase attaches. The operator is a region of DNA on the operon that interacts with a regulatory protein which controls transcription. Translation has to do with the formation of amino acid chains. This process occurs in the cytoplasm and at the ribosome. The steps include initiation, elongation, and termination. Initiation starts with rRNA, the subunit, binding to mRNA. An initiator, tRNA, with the anticodon binds to the codon AUG, which begins transcription. Binding of the transcription components are caused by initiation factors, a group of proteins, and GTP. Elongation begins with codon recognition, and this is the anticodon of tRNA binds to the codons on the mRNA on the A site. Peptide bond formation is the peptide bond between the amino acid in the A site and the carboxyl end of the polypeptide on the P site. Translocation is when tRNA is moved from the A site to the P site, then to the E site, from which it is released. The last step is termination, and this is when the single-stranded RNA molecule consisting of the codons, which are sequences of 3 nucleotides, forms a unit on the genetic code. Post-transitional modification. It occurs after translation. It's when a protein completes extra steps in order to be fully functional, and occurs at the peptide terminus. One example is glycosylation, and is when a carbohydrate is added to the protein. Methylation is the addition of a methyl group to either lysine or arginine. And lastly, phosphorylation is when a phosphate group is added to either serine, tyrosine, or threonine. Prokaryotes have no compartments in their cell. Thus, transcription and translation occur concurrently on the same gene. The newly made protein is able to transport efficiently to where it is needed, as there is no segregation in the prokaryotic cell. However, in the eukaryotic cell, the separation of the nucleus from the cytoplasm allows processing to take place, which does not occur in prokaryotes. This additional stage aids in regulation and coordination. Eukaryotes also contain a crucial mechanism allowing it to transport proteins from one organelle to another. So the genetic code is universal and shared by many organisms. An organism with the same codon will translate the same amino acid, and similar genomes aid in showing common ancestry. The significance of the codes is similar to a common language shared by all living organisms and has been present in all common ancestors of modern organisms. It portrays the relationship between all life on Earth.